afternoon. If maybe we can just have one meeting, please. My name is, as shown on the board, Mpeku Maina, one of the managers in the Soil, Climate and Water Research Unit of the Agricultural Research Council. I will be chairing this afternoon session on the conservation and sustainable use of natural resources. So time is very tight, so we're going to allocate around 20 minutes per presentation, and that 20 minutes include the crossover. So my appeal to people who will be presenting is that they should present for 15 minutes, and at least we'll have about 5 minutes to engage with the presenter. However, we are going to slightly change the program because sadly one of our presenter is not yet here. So we are going to start with the second presenter, who is Doctor, according to this program, Dr. Dave Tanner. Dr. Dave Tanner is a specialist researcher in the soil science program in the Agriculture Research Council. So if maybe Dr. Tanner can, the floor is yours, sir.
Now, can I just press on to the next one? Where is Dostane? Good afternoon, ladies and gentlemen, and I will hope that you can all see me. I have it in front of me, but not yet on the screen. There we are, thank you. I'm going to talk about the viewpoint, or a viewpoint, on soil sustainability. And I thought we'd just kick off a little bit with something about the background of the soil properties, and then we'll look at several aspects of soil sustainability. Obviously the field is very wide, and in the time that we have, we won't be able to look at all of them. I thought here that we'd look at some of the soil properties that are responsible in our eco-services. And we have those properties that are stable and don't change very much over a long period of time. We've heard about indigenous animal breeds this morning, and something about medicinal and indigenous plants. And I'm going to say something about indigenous soils. They've obviously been there for a long time. And the stable properties are that as the soil weathers in place, we find that some of our mineralogy and our texture doesn't change very much over time. They are inherent and important soil properties. We can go down a little bit and we can see some of those properties that are still stable but do change. Cation exchange capacity, pH, acidity are among those. And then we can see some of the very dynamic properties, organic matter. As we till our soils, we might start off with, in South Africa, 2% organic matter will be high. And as we till that, we might find that it's down under 1%. Water content is changing dramatically, minute by minute. And then also the soil biology, and that's probably something that we don't have a good handle on in South Africa, but that's one of the dramatic properties, dynamic properties. And how do these influence the way that we understand soils? Well, we have the macrofaunal. Bacteria and fungi could be as examples, and they are adapted to these soil conditions. We have plant life, and they adapted to also the soil properties. Terminalia trees we'll find in our sandy soils. Acacias will be probably in the valley bottoms. where there's an accumulation of the basic cations. And then grasses might be not so, have a much wider range. These properties, they change slower, and obviously they're going to respond, and their stability and their sustainability would also then be different. We could look at the macrobiology, the larger animals, and they are functioning in with this plant and organic matter system, and giving us also the various dynamics. How do we fit into it all? Well, we as humans, we're thinking about our agricultural and our industrial interventions, and we could think of pristine wilderness, where there's natural long-term stability and hence natural long-term sustainability. Our rangeland and our rain-fed agricultural systems, we're certainly aiming at an economic, sustainable production. It's questionable whether we always do that, but that's certainly foremost in the minds of many of our producers, with a long-term ecological sustainability. So there we're looking at how our production systems would retain that sustainability. Urban disturbance. Obviously we have a natural altered ecology in our urban cities. And the point that I think is important here to make is that we need to be thinking increasingly of what is a green urban environment. And I attended a lecture not so long ago where the engineer, but obviously also in terms of agriculture and in water, spoke about a city in Singapore that had changed its philosophy of the engineering and of the approach to almost a green, what we might call a green city. And I think it's an important message that we in South Africa need to be thinking very seriously about. On our Eastern Highveld, we have coal measures. And there's huge areas, and there will be into the future too, where there's open cast mining. And at the expense of being over dramatic, there's probably disturbance there that may well be largely in terms of a soil time span irreversible. So in our human society, we're interacting with these interventions all the time. The pillars of sustainability, biophysical, and I'm going to talk largely about soil, but also climate and water and others. There's economic sustainability, social sustainability, and I heard this morning from Dr. Moshe Swart what might have been, you might call spiritual sustainability. Let's go on. Something that we in our unit and the Agricultural Research Council are particularly proud about is an algorithm that we've done where we have an underlying basis of our natural resources stemming from a long term where we've been looking at soil and climate and water, but largely soil in my case. And what you can see on the right there is one example of what we've described suitability for maize production under rain-fed maize in one of our provinces, the Northwest Province. And the orange and the brown and the yellow is looking at moderate and higher potentials in those districts. The pink and purple, much lower potential. We have over 40 of these algorithms that are looking at rain-fed and irrigated crops. And the way that it works is that we are taking our soil information, be it soil depth, texture, cation exchange capacity, acidity, all those properties that we characteristically measure in soils. We are testing those against the crop factors. We're looking at something like the terrain and climate, and all of these are integrated and then produces a map like we have over there. That spatial stability then is then better than 1 in 250,000 or better. And what's important about it is that the maps have spatial integrity. In other words, where there's a polygon, we can go back to the original factors of why that was considered to be what it is, and it's not just a something, but something that has scientific importance. So we are quite proud of that. Below down on the screen, there's one for soya and one for irrigated potatoes. Let's look at it at a bigger sense than that. Why do we looking at this stability or this sustainability? And obviously it has an importance in the provincial and in the regional development planning. And our institute has been involved with the Department of Rural Affairs and Rural Development and Land Affairs in looking at, in at least 3 provinces, a sustainability for land suitability for that here. I'll just take you one down, and I think you might see this slide again. The picture on the top is a projection for the year 2015, and at the bottom, a projection for the year 2060. And this is a future scenario. And one of the things that I thought is, where are we now, and where might we be moving to, and what might be the indications on terms of sustainability in soils and suitability in soils that might give us some indication into the future. Obviously, we've used soil properties of projected climate, and I'm going to think more about the soil properties, and you might hear a bit later about the projected climate. And what's evident here is that there's a general temperature increase from the west moving eastwards, and from the north moving southwards. And so if you look at the top picture, you can see what the suitability is, the green and the dark green, projected now. And in that number of years, about 40 years from now, that would have moved towards the east and would have moved towards the south. And so obviously there's a climate change here, but there's an implication as well in terms of the water delivery that we're going to be finding in those soils. And what are we going to do about it? Well, I think there might be some things that we're thinking about, but the first important point is to know that we have this change in the projection there. Our institute also has been very active over a long period of time in looking at detailed suitability assessments. And this one here is a map of a farm, and you can see 4 or 5 different soils there that are mapped out. These have been mapped by a grid process where we've walked grids about 150 meters, depending on the scale that we need and the detail, and looked at and used our classification system to come up with those map units. How do we use it? Well, first of all, if we know that we've got deep, freely drained soils, we're going to recommend that for arable cropping. So that deals there with the soil suitability. If we know that we've only got 300 millimeters on a hard rock, well, obviously those are the ones that we're going to avoid. So it's the best land selection that we're going to be using, and we're going to be telling farmers, and we've been doing it in our institute, and it's done widely in the private sector, of selecting the best land. What's very important about this kind of development that we've been looking at locally is that we're looking at the best rain-fed water delivery, because it's the water that's driving the crop production in these parts. So we have that there. We have recommended crops and cultivars, even cultivars that are coming now, fertilizer and liming, protection and erosion, also coming into important. This slide here deals with suitability in precision agriculture. And because we've been talking in resource-limited farming, I thought it important, and I questioned my colleague before I spoke about this, is that land selection is very important. At the top slide, you can see that there's been good land selection above the trees, and obviously that's not always in many of our rural areas. We have not always chosen the best land to put on our communal cropland, so that is a good example of where it has been done well. Soil analysis, I can't emphasize it more. We have seen that we can use certain cultivars that can take the nitrogen out, but if there is no nitrogen, if there's no phosphate, we cannot expect that we're going to get good yields. So soil analysis, fertility, liming are very important aspects of our land selection and our soil science. Here we've talked about in the bottom picture, and there's many homestead gardens in many of our rural areas, and we've worked a lot there. And so water harvesting and conservation agriculture, which flows from the soil, is an important recommendation. And then integrated support systems, provincial and private extension, and obviously the financial marketing system all go together. And that together as the total package, beginning possibly in my instance with soil, is one of the important aspects. What about our soil analysis? Well, up to now we've used wet chemistry as being one of the important aspects. Extraction solutions, mechanical analysis as flowing from that. But what of the future perception? I expect that we're going to be using wet chemistry for some time to come. But what is important about it? I think we need to improve on the way that our systems are integrated, from the time that the sample arrives at the laboratory, through all the process of analysis, and through the recommendation that goes out to the client. There's still a lot of mechanization that can be automated in those systems. ISO certified procedures, very important. New analysis technology is appearing on the market all the time. And one of the importance of these is near infrared. And what it can do, and there are certain laboratories already doing it, they're measuring organic carbon, clay and water, in the soil, but I believe, according to the literature that I've studied, there are many other, and cations and phosphate could be important. So we could be taking a sample from a very rural area, having it sent to the laboratory, and a very short time, without destructive and a low cost, we could have a reasonable and robust fertilizer analysis program that's taking place. Our laboratories often work on a mass basis. In other words, we're taking the milli equivalents or cation, the centimoles per kilogram of soil. And that's fine, and we're getting a high accuracy with that. But if we also were to think in fertility, where the efficiencies of scale are derived from a volume basis, we can be equally, with a little less precision, but with a higher volume and throughput, we can be looking at a much more sustainable and a farmer recommendation system. And so I present that there as the volume basis as opposed to a mass basis. I am a specialist in soil classification, or I like to think I am, but it's certainly one of my passions. Thank you. I'm glad that somebody could laugh. But well, we have, and I've said it in many public fora, that we have a very good soil classification system in South Africa. You'd see a copy of the binomial and the taxonomy, the red book and the blue book, and they've served our agriculture and our environmental understanding very well in the last time since about 1970. What do? They help us enormously with land selection, evaluation of land, and farms are changing hands, and the value of the farm in terms of the monetary value, the rands, is very much associated with what the soils are that we have there. So farmers and estate persons know about that. It is the basis of our agricultural production systems. And how does it work? Well, it works on the diagnostic horizons. I'm not going to talk a lot about that, but you can see on the bottom there, you can see where we've been looking at over there. What is the future perception? Well, in the international situation, and we follow quite closely with the World Reference Base for Soil Resources, which comes out of Wageningen and Holland, that I believe we are going to be looking more at the recognition of all deep horizons, because that's where our water delivery is becoming increasingly important. There has been a recent paper that's supported by many of the authors of WRB, World Reference Base, and we are following in much the same trend. What will we need to do? Well, we need to revise the way that we look at diagnostic horizons, not in terms of the definition, but not in terms of the concept of what we have there. So that is very important. We need also to have an open-ended system, and we're working towards that, that as we find new soils, we can put them into our classification system, we can attach the limitations that are there, and without disrupting the system. So I see, as happened in 1970 when the Red Book was published, I see somewhat of an explosion of soil information that I believe will develop and help our agriculture very much. I think I'm nearly finished. Here, what is the future of that? Well, we have archive and paper systems that have served us well. But digital mapping will come more and more into the future. So I'm going to skip over that, but I see that the way that we need to have this detailed soil information, because that's where our agriculture can intensify. How can we do that? We can do it by digital soil information. We can do it by advances in hydropodology, which is a new development in our science. We can do it also, and I would like to leave the system here and the thought here that we need to have advanced data capture systems. And there are many ideas that we could use where we're using the modern electronic information systems that could be integrated into our soil systems. So I close off there with an integrated information electronic system, and I thought I'd just show you this here, 4 different soils. The one on the left, or on your left, one of our soils that are stable, that's giving us the water delivery out of our high mountain areas, a swelling black clay. The one in the middle there, the yellow one of our high production soils with water in the capillary water in the grey part of the bottom, and the last one, a solid calcareous lime layer. And that was all that I had to say. There are others, obviously, soil chemistry, soil biology, very important, but I think my time was up. And so I hope that I've given you some ideas of how I think soil sustainability might be moving into the future. Thank you.
Thank you, Dr. Turner, for the detailed prescription of the sort of soil information this country is having. and the critical nature of that information to agriculture. I think the key thing there is that agriculture as it is today, 2015, when you look in years to come, as you say, 2060, there are challenges because production areas might not really be similar. Colleagues, is there any question to Dr. Tanner based on what he just presented? Can I have maybe 3 hands if they are? So it would be 1, 2. Any other hand? 3? Yeah, in that order, please.
Good afternoon. My name is Jacinta. My question to you, sir, is we already now understand how critical soil analysis is to crop production. Now, most of the work that you have done is very useful for scientists and, of course, for farmers. But then, is there a way forward in how we can find local ways of doing soil analysis or locally fabricated methods of doing soil analysis on the farm? Because most of our smallholder farmers will not be able to afford the kind of comprehensive analysis that they need to be able to really do well with their production. So I hope my question is clear, sir. Yes, so I'm saying, moving forward, what can we do in that area so it can be applicable on the field?
I think there are important aspects where we could look at. Firstly, I personally don't think that we should be analyzing the soil on the farm, but I think the way forward might be that we need efficient methods, and that's what I spoke about at the end, where the soil is collected on the farm, a picture is taken, it is transferred to the laboratory very quickly, and with some of the new chemical methods and a robust fertilizer recommendation, I think that we can, within a very short time, be supplying an adequate recommendation. One thing might be to look at all the schools that we have in our rural areas and to be collecting samples from those schools. So yes, I think there are areas, but I don't think that it would be a rapid, you might put a test on the farm itself. I think that the advisor and the recommendation system would be better served through a centralized point. I hope it answers your question. I know it's been a topic that has been much researched in the US, but I think that that's the way forward, and I think it can be done efficiently into the future.
Yeah, can we? No.
This is a very general question, but you alluded to the damage that might take place because of mining of soil, and I just wondered, can you sort of give an idea of why soil will not actually restructure in our lifetime, kind of thing, or what's the consequence of completely turning over and disturbing the soil deeply?
One of the biggest consequences in compaction. What they do is they strip the top off, stockpile it, and a lot of the biological activity in the stockpile gets lost because it's not aerated and it's not watered. The next point is they use very heavy machinery to put that stockpile back so that they're taking the soil back, but we're ending out with bulk densities of in excess of 1.8, 1.9, and plant roots just simply don't get into it. So there are serious limitations as far as what happens is that the land looks good, but its ability to produce is somewhat limited. And it is a challenge, and I do think that as biology takes place, but I don't expect it in our lifetime, that we are going to find that production, which in our Highveld is a high producing area, that we're going to find it now. But yes, in the longer term, it could be. So if we can overcome that compaction problem, and if we can use fertility and organic matter, yes, they can be overcome, but I think it's a longer term action.
Yeah.
Thank you. My name is Mafongoya from University of KwaZulu-Natal. I used to be a very good soil scientist. I'm a South Africa research chair on agronomy and rural development. Now, two things. I don't think you are right, Dr. Taylor, that you cannot analyze soil on the farm. If you know one of the most celebrated soil scientists in the world is Dr. Pedro Sanchez, who was my DG at EPRA. Him and his friend have manufactured gadgets which are called soil dog. With young kids can take, say one youth in agriculture, they can take this gadget on a truck and they can make a fertilizer there. We need to move with that. That's number one. Number two, from Jacinda, we've done work on indigenous soil classification by farmers. And when we do that indigenous classification, dot for dot matches your lab. Farmers know what soil, that the color, what it means, the texture, what crop can they grow there, how susceptible is to erosion and so on. So we shouldn't shut out that there's a lot of indigenous knowledge on soils there, on their classification, their use, just as much as you do with your pedagogy. So I think these are innovative ways. But I didn't hear about you with your rural areas. When I did soils work in South Africa, I've never seen reports on soil mining and depletion, where your farmers are using less fertilizer. What extent are South African soils in rural areas are being depleted? In other words, depletion is attained without pressing. I was reminded by bank manager that Professor Kamp, I want to see because I was taking money without deposit when you're mining. So we want to know about it. Thank you.
The first one I'm not going to answer in any detail. I tried to say I think there are areas that we could be looking at. I think it's innovation, and one of them is that you talked about that innovation. I do feel, and I still feel strongly, that the analysis ought to be done in an adequately provisional laboratory. Now, we saw a truck that went down, that could work, and the system could be in there. So yes, I could go support on that. The second point is I'm a strong believer in the correlations between indigenous and scientific systems. So please, I didn't have a lot of time to do that, but I'm a strong believer in that, and I didn't quite understand your last point, but I feel very strongly that we need to be looking at areas where we can supplement the natural fertility, and there are ways of doing it. Biology is going to be one into the future. We are not well endowed in our research here in biology, but I do believe that we are endowed with an ability to analyze and to supply the fertility there. And that technology and that understanding is there. So it should not be an area where we need to retract in our crop production. I hope that answers some questions, and I'll be here a little bit later and you can challenge me a bit later on. Thank you.
All right. Can we clap for Dr. Turner, please? Yeah, I think for people who just joined us, we started with a second presentation, so you might be a bit confused in terms of where we are. So now we'll be asking the third presenter on the program to really come on stage. His name is Dr. George Chirima. Dr. Chirima is the research team manager responsible for geoinformation sciences in the ARC. Dr. Chirima, the floor is yours.
Thanks, Dr. Maila. Good afternoon, colleagues. Right, I come from remote sensing and GIS. So my talk was not really too much science, but I try to answer the question that people ask me all the time. What do remote sensors do in agriculture? Many people are not very clear on what we do. Well, I'll try and give some of the answers now, and maybe we can expand later. What we probably do mostly is we integrate all this data. You will see that there will be pictures that you have seen and data that's coming from soil sciences, data that's coming from climate. What we do is modeling. We bring that data and try to integrate that data so that it can be useful in a different way. So the good thing with models, you can play around with data and do it, build it and unravel it, build it again without disturbing and creating too much confusion. So that's the beauty about models. So yeah, this slide, this presentation, I would like to acknowledge a lot of people from our unit and a lot of collaborators that we collaborate with. Right. Why is this? How do I move this one? Okay. Right. In that slide, I tried to look at what kind of realities or what kind of space us as remote sensors can be able to operate in. Those are two realities that we are confronted today, that there's not too much investment in agriculture, especially in the rural areas. some of the infrastructure is not there, and some of the people don't know where the infrastructure is. And regions where we need to increase production are the poorest regions of all. And people don't have money, they have a lot of challenges. So us as remote sensors, we try and find the space in trying to help in that area. So these are basically the key constraints that we can narrow down, that the soils, like you've seen, they are already depleted. But we don't know where those soils are. We only know that the yields that are produced, they are very limited. People struggle to produce more than what they can feed in their houses. So that's an indication that the resources are depleted, but you don't know where. We also know that people with those challenges are the poorest. And the basic infrastructure, especially in rural areas, is not there. And this is not a problem for South Africa, it's common for all Africa. But infrastructure is not there. And people don't always know what to produce where. You find people producing maize, the same maize over and over years in the same area, but the yields are not justifying the production. We also look at that people produce, they don't even have a clue or a very limited information of where can they take the excess. How can they access monies to increase production? And of course, when the challenges come, my colleague from climate will tell you later that droughts, a little bit switch in the rainfall pattern, it creates huge problems for farmers. So they are vulnerable to those changes. And of course, sometimes we give people farms and we don't advise them what to do there. Then those farms are not put to proper use. So we call them distress farms. They are farms that need our attention. So that's the space where we think, where our unit tries to contribute, to work across the board and synthesize information, and it tries to contribute with other programs. So the first question, you saw Dave presented some pictures, is on suitability. This question we are like saying, where do you produce? What can you produce in this area? In your area, what is viable? Because if you want to participate in the economy, you need to produce something for yourself and then be able to sell some surplus. So we can put that all information in space and say you can produce here, this is what can be there. So we can guide in terms of what you can produce. And you can also bunch these things in the same model and say, what you are producing now in 2015, can you be producing the same thing in 2019? Like I said, we build models. You can build one, disassemble it and build it again, and then test it to see which is reliable. In other words, what I'm saying here is we advise farmers to say, under this, given these conditions, this is the likely chance that you will be viable as you go forward. So the second part is the advisories. We work closely with our climate guys. We tell you there's drought in your area. This is the performance of your vegetation. It's key for government, for example. They want to know whether there's drought, which areas are highly affected, which areas are people trying to cope. So we can tell them objectively by looking at what's growing in the field. By the way, we rely on satellite imagery. So we can see from our computers what's growing there. Right. So this is similar to what Dave talked about. We can take soil data, and we can ask him, Dave, what are the conditions that maize is growing? And Dave will tell us maize grows in these conditions and so forth. And this is the amount of rainfall maize requires. And this is the growing period. This is the minimum temperature. And then he will tell us if the slope is like that, you cannot grow, or you can grow maize. We put that in a bunch of models, and then you can have pictures and areas that you can pinpoint. You say, okay, in your area, if you grow maize, this is the likely yield that you'll get. And then we can also project this in the future. Those the map that you see there are different models that have been developed elsewhere that predict what will happen to Southern Africa, specifically to South Africa, over 20 years, over 15 years, over 100 years. We take an average of those models, an average performance of those models. We average those models. What they are generally agreeing on is the rate is growing there. It means the area is likely to be drier as you go forward. And then we take that output and put it in together with the Dave soil information and say, Dave, if the temperatures change a little bit with your soils and so forth, where do you think you grow maize? In 100 years, in 50 years, in 20 years? Or do you think it's viable to grow maize now in Mpumalanga? And then you get those products that tell you, yeah, yes, in Mpumalanga you can grow up some maize. But in the other side, you are just wasting time. You're throwing money into the dustbin. So this information is useful for people who are either trying to invest in farmers to say, okay, for this area, maybe you're rather safe growing with livestock, or you are safe growing fruit trees and stuff like that. So that's the information that we give out. So we currently have products with that list of plants or list of crops, and that list is going on as we continue to work, developing products and models, how those products will perform over time. And for this, this slide tells you the things that we can warn farmers that we are capable of developing models to look at crop performance. Is the crop stressed? Do you have enough water in the soil? What happens is plants give out a different signal if there's water in the soil. If there's no water in the soil, they give out some stress signal. So you can pick those up from using satellite images. So you can generate products and give farmers that information to say, yo, plants are stressed. We can also look at disease and pests. There are some pests that can ravage your crops. If the crops can give enough signal that we can pick up from satellite imagery, we can also model those and indicate to you the spread of that pest or the spread of that disease. And so it also helps producers or government to invest in either fighting the spread of those pests. The next slide indicates all this information. We don't keep it in our labs. We try and package it in a way that the public can also access. So this is one of our newsletters that we give out monthly, which indicates the conditions that are in your area. And people are free to download this. It's a free newsletter. So if you are in an area, if you request a copy, you can get a copy of that newsletter and get to understand what's happening in your area. So it tells you, for example, this is a nice picture. You can compare January over years, because sometimes you can, if there is enough moisture in the soil, you can plant. But if the moisture in the soil is not enough, you can't plant. So these products tell us whether there is enough. If you look at 2016, it's all brownish. It indicates there's not enough moisture in the soil. So planting again, you're simply wasting money with your seed. So that's kind of decisions that you can make when you look at these products. This also gives another indication of the general trend of the vegetation of an area. We also contribute to the international world, where we give them conditions of our crops or expected crops in Africa under GeoGram. So we send them information, and so they can monitor and say whether we really have serious problems in Southern Africa and so forth. And they put it as a global product. It's called the GeoGram product. And so we participate in that space as well. And we also have another function which is critical for the country. For planning purposes, we map or we estimate the crop that's grown for the country. This is important for prices. The government needs to know what price. If there's enough maize, the prices are less. If there's very little, the prices go up. So we provide this information to government for planning purposes. They also use the information if they want to import. They want to know how much they are going to import. If they want to export the surplus, they want to know how much they are exporting. So we provide this activity together. We are a consortium. We have private partners, 2 of them, and the ARC. So we map the fields all over the country, and we look at what is harvested there, what is delivered to the grain, and eventually we do some statistical calculations and get the final product. So this is the kind of information that's generated. You can do it per province, per district, and then you can compare over the years whether the production per district is increasing or is coming down, or if you want to make some interventions, where do you make the interventions in order to increase production. Right. We also look at the competing vices. For example, things that compete with agriculture. You are aware that if soil is degraded, if there is invasive species that compete for water, if there is vegetation degradation, generally these compete with agriculture, they reduce the productivity. So using satellite imagery, we look at identifying whether these vices are increasing or they are decreasing, or where is the problem. So we use satellite images to monitor those things. Here is, for example, how we monitor alien invasives. We look at the rate these invasives are encroaching certain areas, and that map over there in the top corner shows areas that are problematic, and this other map shows how we at least look at imagery complementing with the groundwork to look at which places have been infested with invasive plants. We look at soil degradation, especially the big areas in terms where the dongas are developing. If you can pick it up with the satellite imagery, we also show which areas have erosion problems, and then government or the provincial authorities can go and try and rehabilitate those areas. We also collaborate with universities and others, and so what we try to do is automate everything. For example, if we estimate yields now physically, we have to fly and survey, but we want to develop these things into automation. So we are looking at signals that can be automated with collaborations with the University of Pretoria and the University of the Witwatersrand. So these are some of the products trying to pick up a signal that we can automate, and then we can use this to predict yields. And the idea is to predict yields throughout the growing season, because now we do it at the end of the growing season, but now we want to monitor the crop and get information as to whether the crop will be successful or not, depending on the changing climatic conditions. Right. This one is also collaboration that we are looking at. You know, it is very difficult to maintain rainfall stations. They are very difficult. So we try to see if we can use satellite products to calibrate or estimate rainfall quite accurately. And we've been having some positive results, because satellite products are all over the country. They cover a lot of area. As you would recall, that a rainfall station only covers maybe maybe you can't put them 100 meters across. They cover over short areas. So satellite will be probably the best way to go in future. Yeah, this is underground fires that you can pick, especially in coal mining areas. You can pick up underground fires. So we use that also in collaboration with others. Yeah, basically I've given you what we can do for those that are wondering what remote sensing can be doing in agriculture. This is the kind of space that you can help integrate information from the other units and create a product that will be useful to government, to farmers, and so forth. Thank you very much.
Colleagues, we thank Dr. Chirima for highlighting some of the constraints in agriculture in which remote sensing and imaging can really be useful. And also I think for highlighting some of the major projects that his unit is involved in trying to develop very important decision support system for farmers. Is there any question to Dr. Chirima? There is one. If we can maybe take about 3 questions. There's one. Any other question? It looks like you'll be the only one, sir.
Thank you very much. My name is Yarama. I come from Nigeria, ARC. I'm not a soil scientist. But recently I was in a meeting with soil scientists in Abuja on the African Soil Information Systems program. And I do know that several scientists were still insisting the need for soil survey maps. And the understanding I want to get more clarification is, while modeling is doing well, what is the relationship possibly from your studies between data from modeling and actual data from detailed soil surveys at reasonable scales in terms of performances of crops as recommended by these different methods of approach? Secondly, this morning, part of the presentation we had indicated from the maize improvement program specifically, there was an improvement in maize variety that was able to increase farmers' yield, I understand, from 0.6 tons per hectare to 1.4 tons per hectare. And I feel 1.4 improved variety. It's a little worrisome to me. So what is the problem with maize in South Africa? Because I know in other parts of Africa, even at farmers' farms, there are places we have seen up to 7 tons per hectare. And these were maize hybrids, if I understand. So have your soil studies through modeling been able to look into those fields to identify what is the problem? Is it basically the soil, or are there some other constraints that is limiting maize production to 1.4 tons per hectare and is seen as significant improvement? Thank you.
All right, thank you very much. The second question, I don't want to speculate. I think you'd probably need the maize people to be able to, but I can just guess maybe. Because you see, the conditions are different. In South Africa, we were a dry area, I think, because the rainfall is not good. But yes, I would ask you to look at maybe engaging maize and soil scientists for a comprehensive answer on that one. But on the first one, on the models and the soil surveys, yes, I can be able to say. The problem is remote sensing is resolution. That's the first thing. There will be products that are at maybe 30 kilometers by 30 kilometers. So you are looking at a bigger area. So the quality of the data that you get from that product is not the same as somebody who's digging there. Right. But there are products with a higher resolution as well, up to centimeters. But what is important is to be able to get a signal from the pits, the soil profiles, that you can relate to some wavelength on an image. Then you can improve accuracy. Yeah, that's work in progress. You need to look at data from the soils, like what we have now in Dev's project. They have data from the 70s, tons and tons of data. What we are looking now is using that data, correlated to satellite imagery, and pick up signals that you can say, okay, if you use this signal, perhaps this is the kind of information that you can get from the soil. But yeah, it's about resolution that you get from satellite imagery and the resolution that you are getting from the soil pit. So those are the two things that needs to be reconciled. It's not one will replace the other. Obviously, there's value in doing that kind of research because it's cheaper to map the whole of South Africa if you get a good relationship between what's in the pit and on the satellite.
Okay, yeah, this just one last question.
Yeah,
thank you.
I'm very excited about this satellite rainfall.
Yes.
I was told that the density of weather stations in Africa is equal to what they have in Switzerland. So you're right, we don't have enough weather stations.
Yes.
So this is satellite rainfall where you can monitor during the season how is it correlated, like flowing from him, with the daily measure. Are we dealing with noise here? We are dealing with the real phenomena.
Right. Where is this one now?
Satellite rainfall.
Right. This is what you have to do. This is the project that we're running now. We're taking several products from satellites and trying to. So what you do is you pick reliable stations. So they're very difficult to maintain with those stations. So you try and pick data from a reliable station and also try and pick satellite imageries that were flying and that are cloud-free at that station when you obtain. And then try and correlate and see. But the results are promising. They're not bad. If we have time, we can maybe speak outside. We're getting more than 60% correlation in some others. For others, it's more, but it's work in progress. So we need to continue to work and see which is the best product. Because for Africa in particular, besides that the rainfall stations are far in between, some of them are maintained, the data is not reliable. So this is a way, if we can break through this, this is a way in which rainfall data becomes easily accessible. So there is value in trying to pursue this kind of research. But yeah, we have not yet concluded. We think one of the major challenges is the data. Satellite data is expensive. The data that you really want, that is very expensive. So you're trying to balance the expenses and what you can get.
Okay.
Very good.
Yeah, you're making it difficult for us not to recognize you. Okay, ma'am.
Thank you. My name is Makanani Sifunzani from DAF. My question to you, you have indicated in one of your slides the publications that you do, and I tend to see one that you were showing us the availability of moisture in the soil for advisory purposes, especially to farmers. What I would want to know is that before you publish such kind of information, do you also link the information that you have together with the indigenous knowledge that farmers could have? For instance, if I'm talking of a rainfall whereby you'll be advising farmers not to go to start the activities of farming because maybe rain, it's not gonna, it will be above normal. Do you also consider the indigenous knowledge before you publish? Because we make it difficult, okay, we tend to have a difficult situation whereby you find that farmers are complaining that this is your publication, this is your advice, here there's rain, and yet you tell us not to go to proceed with the activities. I just want to know that point.
All right, thank you very much. That's a very important question in terms of how do you tie up what we do with what is on the ground. Yeah, it's important, but I think we have a project that's coming up now that's going to work closely with the indigenous people, and then the information they have will fit in the project. I think Terry is there, they're going to run a project now, but currently we don't consider the information that's coming from the indigenous people. It's just our South African weather station in a network and ARC weather stations that we use for that publication. But clearly, yes, what you raise is important. If we can be more direct with farmers, I think the product will also be widely, and it can be even more accurate. So yeah, it's a point taken in terms of work.
Yes. All right. Yeah, ma'am, further, we just want also to refer you to some of the work that a colleague of ours did, Dr. Guku Zuma Netsiku. I think she'll be glad to engage you around that particular issue that you raised, the indigenous knowledge and how it can complement the sort of information that you were talking about. Thank you. Yeah, I think we can thank Dr. Colleagues, our next presentation is by Dr. Justin Harting. Dr. Justin Harting is the acting senior manager research at one of our institutes, the Small Grain Institute. He'll be presenting something about the importance of biocontrol. Just want to add that he's also going to be joined by our colleague around the same issue of biocontrol use, Dr. Roger Price, who is the wheat and insect control research team manager at our Plant Health Institute.
Thank you very much. Good afternoon, ladies and gentlemen. I've been asked just to give a very short overview. Our presentation has been split into two parts. Myself with Dr. Roger Price, as has been mentioned. So I'm going to just cover the insect pathology side of the work that's being done at the Small Grain Institute in Bethlehem. That's about 300 kilometers from here. So what we're using here is the microorganisms termed entomopathogens. So entomo refers to insects, and pathogens, these organisms that are able to cause a disease in this particular insect pest. So just as a background, one of the major driving forces behind the work that we do is the problem that we have with the development of resistance to insecticides. And as you can see on this graph, from the late 1950s, early 60s, there's been a tremendous increase in the number of resistance cases, also in the number of species of these insects that are now resistant to the chemicals, and the farmers are with their backs against the wall, and many industries are now approaching us asking for alternative remedies, and for this we are now looking at inducing a disease in that particular pest through the use of a bioinsecticide. You can also see that the withdrawal of chemical insecticides is another big issue, one of the other driving forces, because of carcinogenic properties of these chemicals, environmental pollution, sustained residues on crops, etc. The European markets are becoming very strict in testing. The testing protocols are becoming very sensitive, picking up minute volumes of pesticide on crops. So in 2005 we had the withdrawal of monocrotophos, chlorpyrifos for garden use in 2010, endosulfan, aldicarb in 2012. So that's a whole host of very significant insecticides that have been withdrawn rather recently. Then also, apart now from the fact that we have environmental pollution and toxicity in terms of human poisoning and animal poisoning, in South Africa we still have more than 150 cases every year of farm workers mainly becoming intoxicated by these chemicals. But as you can see there, on the 21st of January 2014, it was the official launch of the South African bioeconomy strategy by the Minister of then Agriculture. There's a website that if you want to look at a bit more detail, but it basically boils down to South Africa wanting to promote the use of natural resources and to turn it into an economically viable industry. So the development of bioinsecticides based on these microbial entities fits the glove rather well. Okay, so the strategies that are considered by SGI, the Small Grain Institute, or the ARC as such, when using these entomopathogens, I've just divided it into three basic categories. First of all, conservation biological control, CBC. Here we promote the survival of naturally occurring entomopathogens. In other words, we're talking about ecosystem services, and how can we promote the ecosystem services? They're already there. It doesn't cost the farmer anything, but we need to know the species complex. We need to understand the phenology. In other words, in which time of the year do these pathogens occur? And we need to understand the impact of pesticides. This is a very important one, and I'll show you a little bit of data on that. And this whole principle is actually termed agroecological farming, and we had some discussions about this yesterday in one of the second theme, I believe it was. Okay, so I'm going to just quickly show you a couple of slides on specifically conservation biocontrol. The second one is probably more familiar to you, and that's the development of bioinsecticides. So here we basically follow a chemical paradigm where the entity, let's make it a fungal spore, is mass produced, it's formulated, and it's sold or commercialized as a as an insecticide. In other words, the farmer would spray this using conventional or slightly adapted equipment, and he will suppress the pest. The pest may resurge again, and another application could be needed. So a lot similar to what one would be using a chemical insecticide. Then lastly, I'll just quickly show one slide on the development of bioinoculants, and here I'm referring to the term induced systemic resistance, and here we're using what we call endophytes. It's a microorganism that lives within the plant, and while it's living in the plant, it is now protecting that plant against certain biotic stresses. So if we look at conservation biocontrol, what we did in terms of cereal aphids, we launched some surveys quite a couple of years ago, and we quantified the species complex of these fungi, and I'm referring here specifically to fungi, and I'll also be talking or just mentioning something on insect pathogenic nematodes. There's just not enough time to go into detail on all of these entities, but at the Small Grain Institute we're concentrating on fungi and insect pathogenic nematodes. So in terms of cereal aphids, these aphids are attacked in South Africa by a whole host of fungal entomopathogens, and those ones that have been marked there, those are the species that we found in South Africa. And the two ones in dark font, Pandora nuaphidus and Conidiabolus obscurus, those are the two most prominent or prevalent pathogens attacking cereal aphids in South Africa. We also looked at the impact of these entomopathogenic fungi on cereal aphid populations, and that was a publication that we brought out. And it was interesting to note what the actual impact of these entities can be on an aphid population. Aphids are typically are strategists, so they have a very rapid rate of increase. They, in South Africa, we don't have the sexual form of aphids. It's all through parthenogenetic reproduction. So these aphid populations increase very rapidly, and farmers can suffer huge losses. In the wheat industry, we have the Russian wheat aphid, is our most prominent aphid pest, and this is one of the insects that we try to understand how these pathogens can impact this particular aphid. Right, so coming back to the most prevalent one, that's just a couple of morphological slides of Pandora nuaphidus. You can see on the pictures there, the color photos, you've got those green aphids, those are the healthy ones, and then the brown ones have all been killed by this fungus. The fungus uses a particular structure. It's like a sucker. You can see down on the right-hand slide, the black and white one, that's like a sucker. And this insect cadaver is attached to the host plant through that structure. And the fungus then produces ballistospores. So these spores or conidia are actually actively shot off from the conidiophores or from the fungal structure, and those conidia then infect other aphids, and this can cause a field-wide epizootic within a week. And we noted on the rose grain aphid Metapilophium dorodum, under irrigated conditions, these fungi are so effective that the farmers don't even have to spray for these fungi. The farmers don't know this, but here we have an ecosystem service at work in their wheat fields, rendering an excellent pest control service without any intervention needed from the farmer. So it's really a good benefit to the farmers to have these kind of organisms in your field. Also very important under conservation biocontrol, as I mentioned, is the impact of pesticides on these fungi. We looked specifically at a couple of insecticides, and you can see those just marked in green were actually, you'll see in the first column we've got B. bassiana, that's the fungus, plus a particular insecticide. And that combination, it's marked in green, had a stimulatory effect on fungal vegetative growth. The blue ones were neutral, no positive or negative impact on fungal growth, and the red blocks was an inhibitory effect. So some of these are good and other ones are bad, and it was quite interesting to note that in that particular case we've got Nomirea relay, with that particular chemical. The one was completely inhibitive and the other one was stimulatory. And just the fungal species that differed in that particular sense. So this is the kind of information that we're researching to understand what chemicals can be compatible and which cannot be compatible with these fungi. Right at the bottom, I've indicated obviously the effect of fungicides, as one would expect, is going to be even more severe. Fungi being more close to plants. And there you can see with Conidiobolus tromboides, even at 1,000 times less the field recommended rate, there was complete inhibition by all of those chemicals mentioned. Sportac, Foliker, Alto-Amyl. Those are critical chemicals that are used for rust control in wheat fields towards the end of the season. So you can just imagine the negative impact these chemicals will have on that particular fungus if it's there and it's suppressing aphid populations. Right, then just quickly moving on to the development of bioinsecticides. These are the 7 key steps that one would normally follow. The identification of the problem, the survey and collection of these pathogens, isolation and testing in the lab, confirming pathogenicity through Koch's postulates. Then you would do your bioassays to select your most virulent strains, mass production and formulation, and field trials, obviously to generate data for registration purposes. In South Africa it is quite strict. We have to register all pesticides under Act 36 of 1947, and those data are needed for those purposes. And then obviously if you then have good data, you would go on to marketing distribution. One looks at about at least a 6 to 10 year period from number 1 through to number 7 to get a product on the market. So this is just to show another aspect of the use of these bioinsecticides. Farmers tend to want to measure the impact of a bioinsecticide in terms of mortality. You want to spray today, and tomorrow morning you want to see dead insects in your field. And these insect pathogens just don't work that way. They take longer to kill. But what we do find, that is the typical judgment, or typical what you would expect with a chemical insecticide. Once you've applied it, you want to know when has death occurred, and you want that period to be as short as possible. However, with these pathogens, there's a change in behavior prior to mortality. And during this period, these insects actually stop feeding. And if they stop feeding, they might still be sitting on the plant alive, but if they're not feeding well, then obviously you don't have any damage incurred to that crop. And we looked at a bit of data. You can see, I'm not going to spend too much time on this, but the bottom left graph, the 3-dimensional one, you can see the red bar. That's that's a chickpea intake of the African bollworm after it has been inoculated with these different pathogens. The yellow line is a virus, and then we've got different fungal species. But that red line, you'll note there was complete inhibition of feeding by this fungus, which is an excellent attribute for this particular species. Okay, so typical signs of these infected insects in the field. These are photos of Bouveria bassiana, a very important pathogen also for commercialization purposes. It forms this typical white fluff on the insects. There you can see different beetles and larvae infected with this particular fungus. Another one, Metarhizium anisopliae, has a green color. Very important, also has been commercialized worldwide. Then coming to ex situ conservation of entomopathogenic fungi, we have the South African National Collection of Fungi. It is situated on these premises. As you came in this morning, the buildings on your left with the white buildings with a green roof, that houses our biosystematics, the ARC biosystematics complex, where all of our fungal cultures are kept. And we have a living culture, the PPRI culture. It houses 11,000 species. This is affiliated with the World Federation of Culture Collections. And then we have the herbarium collection, or the dried collection. There you can see the numbers. It's quite spectacular. And if we look at major EPF isolates currently in collection, 786 Bouveria, of which 710 is the species bassiana, and 174 Metarhizium. So quite a number of these insect pathogens are available at this facility for research purposes. Then just something on insect pathogenic nematodes. Normally when you mention the word nematode, the farmers think of the bad guys attacking the root systems of plants. However, there's also a beneficial type, the insect pathogenic one, which lives in a symbiotic relationship with a bacterium. The bacterium is actually the agent that kills the insect and not the nematode. The nematode is only used as a vector. So it carries the bacterium in the soil, the nematode enters the insect and it then regurgitates the bacteria. This kills the insect, and the bacteria then assists the nematode with the production of food as it uses the internal organs as a food source. So it really is a symbiotic relationship between these two entities. What we did was conduct surveys across South Africa, and we now have a total of 184 isolates. This collection is kept at the Small Grain Institute in Bethlehem. And also we have discovered 4 new Steinonema species, Tophus, Innovatione, Betlechemi and Bodolphi. Those were just recently published, new species descriptions on those 4 nematode species. And we've also developed a mass production protocol through the PhD of now Dr. Chima Ramakuela. You may have seen her this morning at the Small Grain Institute stall in the Marquise tent. So we have quite a large number of these nematodes also available for commercialization. This is just a slide of the Bergergen spray tower that we use to quantify virulence when we work with a fungi. It's a closed system. It was developed in France way back in the mid-1950s, but it's still very applicable today. It's very effective, and you can spray your insects, you can quantify your dose, and then you can compare between these different. Okay, I've got to get going. There's another talk on its way. Just one strain, that's the white ones, causing much more secondary fungal growth than the other ones. Okay, that we can skip. And then there is a formulation, a product that we've formulated and commercialized called EcoBB, which we do in collaboration with the private industry. And then lastly, the bioinoculants, the endophytic fungi. There's an endophyte, you can see it growing from a leaf, a rose leaf. But also we tested this against the 2-spotted spider mite, and it was pathogenic. So, okay, this I'm just going to flash. Those are just some ideas as to how we can take these bioinsecticides forward. I thank you very much.